We're continuing our studies in Chapter 11 on carbohydrates, and in this lesson we'll be looking at structural carbohydrates. Remember from our last lesson we looked at fuel storage forms. In this lesson we want to look at polymers or carbohydrates that serve a solely structural role. The first we look at is cellulose. This is very common in plants. It's a glucose polymer, and just like amylose, the linkage is a 1,4 linkage. The difference is it's a beta link instead of an alpha link, and that's pictured at the top of the screen here. So again, it's 100% glucose. It's still a 1,4 linkage, but in this case it's beta rather than alpha. And just that simple difference makes for a very profound difference in structure. Instead of compact granules, remember that's the storage form, we get extended fibers, and this gives rigidity and strength for plant cell walls. Those cellulose fibers are pictured at the bottom of the screen here. Very intricate network, very rigid, very strong, and just that linkage is beta rather than alpha. That's the only difference between this and amylose. There's also an extensive hydrogen bonding network that forms within and between chains, and that's illustrated at the bottom of the screen here. So here's the hydrogen bonding network within a chain, and here you can see between the chains here. That also contributes to the rigidity and strength of this structural carbohydrate. Most animals don't make cellulose, and most cannot digest it, and that of course includes us. But microbes do, and so ruminants like cattle and sheep usually contain these types of microflora in their gut, and that allows them to digest the cellulose component of the plants that they eat. Another type of structural carbohydrate is referred to as chitin. Now, just like cellulose, these are beta 1,4 linkages, but the difference is it's not glucose, it's a glucose derivative. In this case, N-acetylglucosamine. So here's the glucose cyclic structure. Our N-acetyl group is in green. Because at the number 2 position we have an amine group rather than an OH, that makes it glucosamine, and we've attached an acetyl group to that amine, and that makes it N-acetylglucosamine. So again, the same beta 1,4 linkages that contributed to the strength of cellulose, and now we have this modification, this derivative of a sugar. This is part of the exoskeletons of insects and crustaceans, a very rigid, very strong structure. It's also the part of the cell walls of many fungi. Another type of structural carbohydrate is a biofilm. It's an extracellular matrix of carbohydrates. In other words, it forms outside the cell. It attaches to surfaces and harbors a community of microbes. They may all be the same type of microbe, or they may be different types. The microbes themselves produce the carbohydrate biofilm, and they maintain it. It is a combination of glucuronate and N-acetylglucosamine, gives them structure and stability. It's responsible for plaque on teeth, and we have a pretty bad case of that in the illustration, the picture on the bottom left. Biofilms can also form on medical implants, and so it does have a medical significance as well. For the microbes, it gives them barriers that protect them from antibiotics and immune cells, and so it's often the case that biofilms form by pathogens that invade our bodies. You can see a colony of a biofilm that forms on a plate on the bottom right. In our next video lesson, we want to see how we can modify proteins with carbohydrate groups, and we'll see the purpose of this glycosylation.